Hello, this is Dr. Divya Pradhan, and today we are going to talk about the scientific process. In other words, how do all the laws or the established theories in biology come about, in science come about, okay? So, first and foremost, you need to understand that biology is, of course, the study of life, and life is such a big thing to be understood easily. So obviously, natural phenomena are being discovered each day. New species are being discovered each day. New things are being discovered each day. And scientists, biologists are scientists basically, and they tend to be curious. So obviously, they are always trying to find out how the world works. So obviously, the first thing about science is, or for biology, is that observations are being made. Okay, and these observations often are not only because of curiosity, but because of the need. If you remember the chaos and the entire confusion that was created when there's an outbreak of Ebola, or new diseases like the Nipah virus. So these things have require scientists to go and find out and see what will work. So some observations are made. For example, let me tell you, the observation that I have made is that by drinking hot water with lemon first thing in the morning on an empty stomach helps me feel very energized. All right. So that is the observation that I have made. So based on that observation, hypotheses are made. So what's a hypothesis? A hypothesis is an educated guess, a tentative reasoning that you've come up with. But before this, let's see another term, and that's called inductive reasoning. So based on the observation, based on the reasoning, based on the reading, based on some of the earlier readings that you've come across, or hearing things, you combine all that information, do some kind of creative thinking, and you combine and come into a cohesive whole statement, and that would be, of course, your hypothesis. All right. Now, to test our hypothesis, of course, now we have to do deductive reasoning. Remember, inductive reasoning is because you induce from some other work. In deductive reasoning is, once you have a hypothesis, you are going to design an experiment, and when you design that experiment, once you get the data from that, you will do deductive reasoning, right? You will use the if-then logic. So, for example, if indeed drinking hot water with lemon boosts your immune system, well, then I should not get sick when others are sick. You see what I'm saying? So that would be deductive reasoning. But wait, first we have to design our experiments, all right? So when you're designing an experiment, obviously you have to have an experimental variable. So experimental variable, in my case, remember, would be drinking hot water with lemon, right? Responding variable, well, what did we check? So we are going to change this variable and test it to see if indeed there's a change in my energy level, or immunity boost, or blood pressure rectification. So these are variables. Another example, well, if I change my shampoo, will my hair shine better? So what am I changing? My shampoo. And what is changing in response? Well, my hair quality, right? Control would be the placebo which is not exposed to the experimental variable. So again, experimental variable is the factor that you are testing. This is the factor that you are testing. Responding variable is the result or the change, because remember, we are just testing. If we are changing the shampoo, we are testing for the hair quality, luster, shine. Okay. Control is not exposed, the group that's not exposed to the experimental variable. Very important. All right. So here, experiments, they have to be in two control groups. One is the one that will get the factor of the experiment is being tested, and here the other one, which is the control, is so here you will see the result of the change that occurs due to the experimental variable. So this is just the same slide, independent and dependent variable, and you understand what the control group is. Okay, this is a repetitive slide. All right, then often they will use models. Some use the fruit fly, some use the mouse. No longer now you can use primates. Earlier, long ago, they used to use primates, you know that. But so these are two very common models that are used in the lab. And then, of course, you also have now computer modeling. So, and this is not only to study climate change, as your book says, but computer modeling would be, to study a lot of other things. You can create your models, and you can do the if-then model of using computer modeling. All right. So after you do this, run this experiment, you are going to get data, right? And this data can be in the form of tables, it can be in the form of graphs. So basically, this is what you are going to collect your data, and then you are going to analyze your data. And for this, we are going to use statistics. So some two terms that when we use statistics is, what is the standard deviation? What's the standard error? This will measure the uncertainty of a value. And then statistical significance. So whatever result you are getting, is it significant? Is it due to chance, or is it because of some other factor? Well, so when you find a result that's significant, everybody gets really excited. Scientists get very excited, because it simply means is that, are you confident that it's real? Is it just that you are lucky just by chance? Were you lucky? Were you unlucky in choosing the sample? Or is it really, truly significant? So obviously, whenever scientists get something of statistical significance, obviously that would be very exciting for them. So say, for example, I found a drug, a medicine to control blood pressure. And if it's statistically significant, hey, that means what? Patent, money, etc. All right. So then, of course, it is published in scientific journals, right? So here, remember, now if you have, like we said, let's go back and let's explain this one more time, is if your hypothesis was statistically significant and it is indeed proved, it's proven, then of course it becomes a theory. All right. If your hypothesis is not significant, then what happens is it is rejected. All right. Say, for example, if I find that the medicine to control blood pressure did not work, then obviously I cannot make it or say indeed that the medicine controlled blood pressure, or hot water controlled, or hot water boosted my immunity. All right. So here, so basically there are lots of theories, the cell theory, homeostasis, evolution. We'll talk about all these terms, but basically the cell theory is that all living things are made up of cells. But like I said, science is changing so fast that now when we talk about prions, hopefully we'll talk about this sometime later. But prions are basically rogue proteins, okay, proteins that have gone rogue. And basically, yeah, they are surviving on their own. They are causing diseases. Remember the mad cow disease by prions. So here no longer is the cell theory really holds true, or we can say the cell theory holds true, but there are many, many exceptions. A good example would be a virus. What would you consider this? As a cell? As a functional cell? No. Or some people consider it as a living cell. Well, don't know. So evolution is something which is highly debatable, changing all the time. So let's give an example. An example would be, that ulcers are caused by a bacterium, and this is the Heliobacter pylori. So antibiotics are used to treat this condition. And so let's see if there are, which of the two antibiotics would I test them. So let's see that I have an antibiotic A and an antibiotic B. And of course, remember, I have to have a control group. Okay. So my hypothesis is that I have discovered a new antibiotic, which is B, and it is better treatment for ulcers than antibiotic A, all right, which is in current use. So my prediction will be that B has to be better because I have done so much of research that this is a new thing, and I have consolidated so much of previous information. Okay, all right. So here I'm going to design my experiment. Control group receives the placebo. That means it does not receive any antibiotic. Group A receives antibiotic A. Group B or another group receives antibiotic B. All right. Now let me see. I'm collect my data over a period of time, and after 2 weeks, I'm going to collect the data, the stomach and the stomach lining, intestinal lining are examined, and if the ulcers are still present, and therefore I collect the data, and then I start putting it in this format in a form of a graph. And here is my control group, here is my test group, here's my test 2 group. Well, so what do we see? So here, look at this. 10% of the people in the control group no longer had ulcers. So the number percent treated, 10% were treated. Okay. 60% with getting the antibiotic A were treated. So no longer had ulcers. 60% no longer had ulcers. But those receiving antibiotic B, 80% were healed, 80% were treated. They no longer had ulcers. So what can you conclude? Of course. So the conclusion is that the hypothesis, that what was our hypothesis? That antibiotic B is more effective than antibiotic what? A, right? So this is a sort of a mini experiment that we conducted. Of course, in order to patent it, it has to go through long processes, many observations. So this is a mini experiment that was designed just to explain the scientific process. I hope this was clear to you all, and if there are any questions or concerns, please feel welcome or to come and talk to me and see me in my office. Yeah? Thank you and have a great day.